Let's get started. Thanks for coming. My name's Sam Ryder, and it's my pleasure to introduce Anitha Pasupathy, who's coming to us from University of Washington. Anitha, something's blinking. That's your laptop? It's like a bomb. Did her PhD at Johns Hopkins and postdoc work. During her PhD, worked on non-human primates and their representation of images in area B4. Then moved on to postdoc in MIT, where she worked on visual learning, and then opened her own lab at University of Washington in 2005 or 6, and has been working on visual processing of shapes and textures, mostly in V4 of non-human primates. And it's a pleasure to welcome you to OIST.
Thank you. Okay, good afternoon. Thank you, Sam, for that introduction. And I want to start by thanking Jonas and Haruka for all their hard work and making all of this possible. It's been amazing 2 months for me, talking to Sam and Yukako about wonderful science, but also enjoying Okinawa on the beaches. So today I want to tell you a little bit about the primate visual system and also some of the work going on in my lab, trying to think about how this part of the brain, the visual system, processes visual information. So please stop me as I go along if there's anything that's unclear, or if you'd like to discuss anything, please just stop me. There isn't really, I don't have to complete any amount of material, so I'm looking forward to a discussion. Okay, so when we look at a complex visual scene such as this one, our primate brain, our visual system, immediately parses this visual scene. We're able to see various objects, the people, the hats, the boats, but also from the textural qualities, we're able to process the stillness of the water, the clearness of the sky. The primate brain is remarkable at this sort of scene understanding. We're able to process scenes even when the information is impoverished. So for example, if I showed you this image, many of you may be able to see an animal in there. I see some nods. I see some puzzled looks. So if for those of you who haven't spotted the animal there, all I have to do is prime you a little bit. So the animal's right there. It's a Dalmatian dog with a collar, his ears, limbs. And now, if you've seen this now, then you've learned it for life. It's one-shot learning. Our primate brain is really remarkable at this sort of scene understanding. And what is really amazing is that the visual system is not acting like a simple camera. It's actually trying to process the scene and trying to understand what is the image that produced that image on the retina. So the image on the retina is a combination of the properties of objects in the world and also other extraneous factors like illumination, your viewing angle, other objects in the scene, etc. So your visual system takes into account all of that context to process this information. And the best way for me to illustrate the importance of context is by showing you a couple of more illusions. This is one that was created by Ted Adelson. It's a brightness illusion. And if I were to ask you which one of these squares is brighter, A or B, I think there'd be universal agreement that B is brighter than A. But if I were to use a light meter and measure the light coming out of this point, this part of the image versus this part of the image, it's actually identical. So I can illustrate that by taking this square here and moving it over there, and then moving that over here. Okay, I never get bored doing that. It's just remarkable how your brain basically interprets this square as being brighter than this, even though the information coming out of that patch of the image is identical. And that's presumably because your brain says, okay, this patch B is in the shadow of the cylinder. And so if the information coming out of this patch versus this patch is identical, then this must be brighter than that. Okay, this is an interpretation of the retinal image. Here's another one. This is also fun. So the way I perceive it, and hopefully you all perceive it the same way, is that this is a big monster chasing this little guy down the hallway. But if I were to give you a ruler, in fact, these two images are identical, right? If I give you a ruler and you can measure this, they are the exact same size. But because of the context provided by all of these lines that are getting, converging, your brain says, okay, this is a long hallway. And if both of these are subtending the exact same visual angle, then this guy must be much bigger than this guy because he is way down the hallway. Okay, so this is the sort of information that contextual information that your brain uses to process visual scenes. We have absolutely no clue how the brain does any of this. It's fun to see this, but we haven't figured out how your brain is processing all of this information. But my all-time favorite visual illusion is this one. This is the Bregman's B illusion. When I look at this set of yellow shards lying there, they don't cohere into any sort of a unified picture for me. But the minute I overlay an ink stain on top, so the yellow bits here and here are identical. But the minute I overlay an ink stain over it, they all cohere into a set of letters B. Okay, so this again is the brain using contextual information. Here, these are just bits lying around. But the minute you partially occlude, you provide some contextual information of partial occlusion, your brain makes up a story about what's out there. Okay, all of this is based on processing along the ventral visual pathway in the primate brain. So what I'm showing you here is the macaque monkey brain. This is the lateral view of the monkey brain. Information from the eye. The first stop for that information is the lateral geniculate nucleus. Then in cortex, the first stop is area V1 in the back of the brain. Then it goes to V2, V4, and then the subregions of inferior temporal cortex. Lots of studies over decades have revealed a lot about how information is processed along this ventral pathway, mostly about area V1. Okay, a lot of the work that's been done, a lot of what we know about processing along the ventral visual pathway has been done by Japanese neuroscientists. So I'll highlight some of that as I go along. One of the things we know is about retinotropy and cortical magnification of area V1. So what is retinotropy? That basically tells you it relates your cortical tissue with the visual field. So which part of the visual field is processed by which region of visual cortex, primary visual cortex in this case. Okay, so lots of work has been done to map the visual field shown here onto cortical tissue. And we also know that the central visual field, so right around where you're fixating, that is represented by the fovea. That part of the image is mapped onto a much larger chunk of primary visual cortex compared to what's in the periphery. So if I were to divide up the visual field into these little squares, then the squares around the point at which your gaze is fixed, that takes up much more of the tissue compared to all this other peripheral space, which is relegated to a much smaller region. So that's called cortical magnification. And both of this, retinotropy and cortical magnification, has been attributed to Gordon Holmes, but really the first person who did all of that work is Dr. Tatsuji Inoue, who was a Japanese ophthalmologist, and he was hired by the Japanese government just after the Russo-Japanese War to evaluate soldiers and their vision loss. And their interest was to give them, to match their pension amounts to the extent of vision loss. And a lot of soldiers were coming back from this war with vision loss, but not, but they were still alive. And this is because of, I don't know the specific details, but there are these new rifles that the Russians were using, and the bullet speed made it such that it caused damage, but it did not kill them. So it didn't it just passed through and caused like very local damage and did not damage too much of the rest. So what Tatsui had to do was just map out the extent of their vision loss, but he went way further and mapped the vision loss to the size of their wounds and the location of their wounds. So basically taking the skull damage patterns, so like the entry and exit wounds, he mapped out where the tissue was lost, and then figured out where the visual field loss was, and then devised an instrument called craniocoordinometer. I imagine it was something like the stereotax to sort of match different cell sizes, and based on that, mapped retinopathy and cortical magnification. And he published all this in 1910, like 10 years before Gordon Holmes did. But a lot of papers still attribute all of this to Gordon Holmes. And then after many, many years later, in the 50s and the 60s, Hubel and Wiesel sort of mapped out what are the properties of these neurons. And so they showed that neurons in B1 are sensitive to the orientation of a visual stimulus. So here they're showing different bar orientations, and the ticks, the vertical ticks here, are the responses of the neuron. So the neuron doesn't respond when the bar is of horizontal orientation, but responds quite briskly when the orientation is vertical. So they mapped neurons in visual cortex, first in cat, and then in the monkey. And people who came after him then showed that this was, in fact, this can be parameterized in terms of tuning for orientation and spatial frequency. And then, much, much later, with lots and lots of work, we now have good models for processing in B1. So we have good models for simple and complex cells in terms of orientation and spatial frequency, and also contrast gain control, luminance gain control, surround modulation, contrast adaptation, et cetera, et cetera, et cetera. Okay, so we have good models for processing at the level of B1. Now, what happens beyond B1? That has been really, really challenging for us to discover. And there are lots of reasons for this, and I'll highlight a few. So basically, the space of all possible shapes is huge. So, if you want to think about what happens beyond B1 and you want to conduct some experiments, you need to choose a subset of your stimuli. But the possible set of stimuli is millions and millions, and where do you start? And you don't know what the important dimensions are. So all of these stimuli can be parameterized in any of many ways, and it's unclear what the relevant dimensions are. So the space is huge, we don't know what the relevant dimensions are. Neurons can be noisy, so you need multiple repetitions for each stimulus. You have a limited amount of time with any given neuron, maybe 1 hour, 2 hours. If you're very, very, very lucky, 4 hours, right? So you have a limited amount of time with neurons, and many of these neurons are tuned in a very high dimensional space. So they're not just tuned for orientation and spatial frequency, they may be tuned for color, they may be tuned for texture, they may be tuned for motion direction of a stimulus. And so the dimensionality can become high, and the interaction between those dimensions can also be nonlinear. So all of these makes trying to figure out what these higher brain areas are doing very, very difficult. Okay, I'm not trying to make a whole bunch of excuses for why we haven't discovered anything. It's just to sort of set up the problem of how difficult this is. So, progress has been slow. Many labs have discovered a lot, and quite a bit of it has been done by Japanese neuroscientists. So let's start by thinking about the highest stages of processing. So the inferior temporal cortex. At the tip of this is the anterior IT, and studies in the 70s and 80s demonstrated that neurons in these higher stages are sensitive to complex stimuli like faces, hands, etc. And a lot of that work was done by Keiji Tanaka at the Riken Institute. And what they did, so the first studies were basically identifying what is it that these neurons responded to. And so they found that they are responsive to faces and hands. And then the question was, what about the faces and hands do these neurons care about? What is the representational basis? Is there something systematic? And a lot of that work was done by Tanaka. And the way they did that work was, so they went to a corner store and bought a bunch of stuffed toys and plastic, fruits and vegetable models, and they put it on lollipop sticks or bigger sticks in this case. And then, so these were anesthetized animals. So the monkey is anesthetized, and they're recording from neurons in the brain, and the eyes of the animal are open, and they're waving these stimuli in front of the animal's eyes. So the reason why they went here was because, the stimuli that were used in V1 and LGN, dots and bars and stuff, were not driving these neurons very effectively. So they figured, okay, it needs to be colorful, it needs to be complex. So they chose a whole variety of stimuli. And then once they identified a stimulus that the neuron responded to, so a lot of times they would have to go through hundreds of these stimuli before they found a stimulus. So this is a very sparse responsive neuron. I mean, the dedication of these scientists is incredible. So they're showing 200 stimuli or 400 stimuli, and one stimulus might drive the responses of this neuron. So they take that stimulus, right? In this case, it's the tiger's head. And then they say, okay, what about the tiger's head does this neuron respond to? So they take away the color, and then they start this process, what they call the reduction method. So they take away attributes of the stimulus and ask whether the neuron still responds to it. And so here the arrows mean the neuron's still responding as they continue to reduce the stimulus. And they get to this point, and the responses are still robust, and any further reduction didn't drive the neurons. And so this would be the critical feature of that particular neuron. And they went through and did this for every neuron that they encountered. And they did this, so here's another example. They started with a screwdriver, and the most reduced stimulus was a hot dog on a stick sort of stimulus, so like an ellipse attached to a vertical line. And I can show you many examples of what they did. And so these are reductions. They started with this particular stimulus, they ended with this. Sometimes you can wonder what the path they took was that brought them there, but it's I can't answer that question. But yeah, so they did this. Yes.
Sorry, I just have a quick question, if that's okay. On the topic of the kind of the reductions and they kind of smoothly-ish transition from the full-on real object to the simplified version, is there any chance that because the neuron would kind of be repeatedly exposed to similar stimulus, so it's possible that it's kind of what it's representing for would drift?
Yeah.
Of course, we can't really do it the other way around, but I'm just wondering.
Yeah, so that's a great question. I mean, there's a question of drift, but there's also the question of adaptation. And you should talk to Dr. Yukako Yamane, who's sitting in the audience, who's the master of this sort of genetic algorithm stuff that she did in IT Cortex. So sort of thinking about, like if you expose the neuron repeatedly to the same stimulus, would the response of that neuron to that stimulus get diminished, and things that it did not see might get relatively enhanced, and therefore you might have trouble. Certainly, that's definitely possible. And so in more recent automated genetic algorithm type versions, you actually, in every generation, you add new stimuli and you mix in different variants of the stimulus so that the neuron's not seeing that same stimulus over and over again, and the effect of adaptation is reduced. So, yeah, thanks for that question. Okay, so they looked at responses of neurons in IT, posterior IT, V4 and V2, and they came up with this this diagram for critical features driving neurons in these various areas. And based on this, they concluded that, you know, in the highest stages of IT, the features are much more complex than in earlier visual areas. It's a tricky conclusion looking at this, but you know, you have to take into account they spent there's a lot of what they learned and what they observed that doesn't go into a simple diagram like this. And so their conclusion reflects things that are not in the paper, that are not in these figures as well. But so and work after Tanaka's work in the 90s. Now we have even firmer understanding of what's happening in IT cortex. We know that neurons in IT cortex are sensitive to 3D form, and this is work done by Yukako when she was a postdoc in Ed Connor's lab. This is some beautiful work using genetic algorithms. And then there's also work again done in Keiji Tanaka's lab by Uzbek Kiani when he was a grad student there. Or a research tech there, I don't remember, but basically showing that at the population level, neurons in IT cortex, the representation is categorical. So for example, if you take a whole bunch of stimuli, so human faces, animals, you know, fruits and vegetables, man-made objects, etc., and then you represent the responses of the population of neurons in a low dimensional space, then you can see that all of the faces sort of cluster together, animals cluster together, etc. So there is something about a structural code, what they call the categorical representation. And then in using human fMRI, there have been several studies that have shown that there are clusters of neurons that are sensitive to faces or bodies in human visual cortex, and these are repeatable clusters that have been, you know, 6 clusters or so for faces that have been repeatedly identified across subjects. So we're sort of learning a little bit about what the representation is at the level of IT cortex. My lab itself is focused on processing in area V4, which is an intermediate stage along this pathway. And we're interested in trying to understand what are the representations here and how do you go from orientation and spatial frequency representation at the level of V1 to higher level form representations in IT cortex. And just like other labs, we've also started with isolate single stimuli, you know, sort of simplified versions of visual stimuli, isolated objects, isolated patches of textures. And the current understanding about area V4 is that this may be the first stage where you're going from a surface-based representation. So at the level of V1, I said the representation is of local orientation and spatial frequency. So that's a surface representation about the characteristics of a surface. From there, you transition through V4 and get to a higher level representation of objects. And we think now that maybe it's at V4 where you start to make that segmentation, where you're starting to pull out objects from the surrounding scene. And neurons here at this level, they encode information about isolated objects using a part space code. What I mean by that is they're encoding information about the shape of the boundary, which can be parameterized in terms of the curvature of the boundary. So for example, this is an example neuron that encodes information about the curvature of the boundary to the upper right of the center of a shape. And so this neuron responds strongly to shapes that have a sharp convexity to the upper right and respond weakly to shapes that do not have that feature. Similarly, these are neurons this is one neuron that responds strongly to shapes that have a concavity at the top of the shape, but respond weakly to shapes that do not. So then neurons like this then together can provide a complete representation of the shape. So if you have a shape like this, neuron 1 will respond strongly because it has that, the shape has that preferred feature. Neuron 2 will respond strongly because that shape has that preferred feature, and so on. So together, a group of neurons can provide information about the curvature all around the boundary of a shape, okay? And V4 neurons are also sensitive to texture. And so this is a very difficult slide to process, so I'll try to walk you through it. So what we've done is we've tried to understand the representation of texture by showing lots of different textures to these neurons and trying to understand, is there a code? And we found that neurons in V4 seem to be encoding information about its perceptual texture dimensions of coarseness, directionality, regularity, and contrast. So in the responses of this is one particular neuron, and these are the responses to different textures. And if you look at just these curves, this is the difference in response to coarseness. So in red are the responses to all the coarse textures, and blue are the responses to the fine textures for this particular neuron. So this neuron responds strongly to all of these textures, which are coarse, and poorly to all of these textures, which are fine. And similarly, it also responds strongly to all of these textures, which are non-directional, compared to all of these textures, which are directional. They all have energy in some particular direction. It doesn't matter which direction it is, but there is some energy in a particular direction. And this is a neuron that doesn't care, that doesn't want textures to be directional. Okay, so we have neurons in V4 that carry information about the characteristics, the perceptual attributes of textures as well. So this is by way of background. And the kinds of questions we're working on right now is to try and understand how do we go from, representation of objects and textures to encoding entire visual scenes. So that's where we're headed. We also want to understand what are the computations that give rise to the transformations from local orientation to higher order shape selectivity. What is the functional architecture? How are these neurons arranged? And then ultimately, how does this contribute to our ability to parse visual scenes and recognize objects? I'm not going to tell you about all of this today. I will just try to get to the first and the last question. Okay, so I'm going to pause just a second to see if there are any questions I can answer. Okay, all right. Moving along. Okay. So this question about how are visual scenes encoded? That's a complicated problem, right? So we've just tried to get some fix at understanding how isolated stimuli are represented. And we're trying to make the next step towards understanding how a little more complicated scene is encoded. And not many labs have done this. And so to sort of help us navigate this path, we've looked to visual psychophysics, because psychophysicists have done millions of experiments. And so, sort of trying to use their results to help guide what we do is something that can be valuable. And so we were looking at studies on crowding. So crowding is a phenomenon where when you're trying to recognize a visual stimulus, when it's isolated, let's say you're looking here and it's isolated over here, you don't have a problem. Your visual system is really good. Regardless of where the stimulus is, you're able to recognize it. But the minute you put other crowders around it, the minute you put distractors around it, then your ability to discriminate that central stimulus becomes very, very difficult, especially as you go further out into the periphery. And this phenomenon is called crowding. So there's a lot of literature on this. Is decades and decades of work. I'm going to reduce it down to one slide here. So basically, let's say you are fixating on this cross here, and you're trying to figure out what that central letter is. That should be relatively easy for you to figure out that's the letter A. Now, let's say you're fixating over here and you try to figure it out. That must be a little bit harder. Okay, so what psychophysicists have found is that, clutter around a target impairs your ability, and clutter that's closer to the object is worse than clutter that's further away. And that distance between the central target and the surrounding clutter scales with eccentricity. So if very close to the fixation spot, so if you're fixating here and you're trying to figure out what this is, the clutter has to be really close to that target for it to sort of interfere with your ability to tell what it is. But if you're fixating over here, and this is in your periphery, then even clutter that's far away impacts your ability. Okay? Yeah.
Isn't that just I have bad peripheral vision?
So if it's isolated, you don't have any problem at all. Mm-hmm. It is only when you start to put clutter. Okay? So yeah, it is, Rosenholz and others have done tons of experiments to show that it's not, you have good resolution. So let's find a size that works at that periphery, and then you put clutter around it, your ability to look at it, to figure out what it is, gets diminished. Okay, and so based on this and lots of psychophysical experiments, people have come up with theories for why this happens. And one attractive theory is the idea that what a neuron does is, each neuron has a receptive field. Now, if there is just one stimulus in the receptive field, it's just encoding the properties of that particular stimulus. When there are multiple stimuli within the receptive field of the neuron, what happens then? Well, one idea is that the neuron encodes the summary statistics of everything that's within its receptive field. So it does not know about segmentation. We talk about segmentation. We can pull objects out of, out of texture. But the neuron, all it does is encodes information about the summary statistics. And so this is, these are psychophysical experiments where what Simoncelli and colleagues did was they had subjects fixate on a spot, and they took these images and replaced patches of the image with scrambled images, which have the same summary statistics, first and second order statistics. And they measured, so that this is what that looks. So this is the original image, and when you replace it with second order statistic, it destroys the boundaries of objects. It maintains the first and second order characteristics, but destroys boundaries of objects. And so they said, well, we did psychophysics on human subjects, and we found that the human subjects couldn't tell the difference between these. And therefore, and they did use different sizes of these windows over which they computed these summary statistics. They said the size of a V2 receptive field is the size at which the scrambles really impair your ability. And therefore, the model is V2 neurons take everything that's within their receptive field and just encode it in terms of the summary statistics. And this is an attractive theory. They supported it with psychophysical results, but there is not incontrovertible proof. So there are studies which argue against this possibility. And this is nice work by Herzog and colleagues, and what they called uncrowding. So the upshot of the theory that I just presented you is that the more clutter there is, the worse you're going to be, right? The longer the line elements, the more number of things there are, you're going to be worse off, because there's the statistics are going to, you can create images that match that statistic. In this experiment, what they did was they had subjects do a Vernier task. So you have two line elements, and you have to tell whether the top line is to the left or right of the bottom one. And they varied the distance between the line elements to measure what is the minimal distance that a subject could still discriminate accurately. Now they started by adding clutter around these elements, and they found that the threshold went up. And when the line elements were longer, to match the sum of the length of the two elements that are of that are the target, then the threshold went even higher. But when they made the lines even longer, the threshold dropped down. So basically, by increasing the length of those lines, they've uncrowded. So they've rescued the crowding effect. And their argument is that by making these lines really long, your visual system is able to group it and discount it. So you've now given the visual system a method to discount these line elements. And so the visual system is able to sort of access this information about these vernier lines and able to do this task. Okay, and they did many experiments like this, and they call this uncrowding. So the idea behind these studies is simply that it's not just some pooled summary statistics, the structure matters. So if there is some structural element where the crowders can be grouped away, then your target still is uncrowded. You don't have the problem of crowding. And so you need to think about more of much more of a segmentation model that's just not sort of that ignores global structure. Okay, but physiologists haven't looked at this question at all. So we wanted to go in and ask, how do neurons in V4 respond to these sorts of stimuli? So the first thing we did, okay, so these are the questions we set out. When there are multiple stimuli within the receptor field, do neurons really encode? Can you explain the responses in terms of summary statistics? And does the encoding model depend on the characteristics of the target stimulus? So if you made the target stimulus somehow more salient, is there more or less crowding? Can I manipulate the level of crowding by just tweaking the characteristics of my target stimulus? Okay, this is all work done by Dr. Taejun Kim, who's a senior scientist in my lab. So the first task for us is to come up with a stimulus that could sort of get at this question. And so we started with our usual stimulus design. So we started with two-dimensional shapes presented either in isolation or surrounded by a set of stimuli that provide clutter. And we varied the distance between the central stimulus and the surrounding clutter. So here they're very close to the central stimulus, here they're far away. And we looked at the responses of the neurons. So in this red line shows the responses of the neuron, of a single neuron in V4 now, to this stimulus, to different orientations of the stimulus. And so it basically says that this neuron shows nice selectivity for that particular shape. Now, as we add clutter around, I'm sorry about these lines, but this gray line here is the responses when the clutter is far away. So you see that the responses go down a bit, but the selectivity remains. When they get a little bit closer, that is this second line over here, it goes down a bit more. And when they are really close, your shape selectivity basically goes away. So this neuron is sort of mimicking the perceptual crowding effect. So as you add clutter close to it, the neuron doesn't seem to know what the orientation of that stimulus is. Okay. Now, similarly, we also did an experiment where we changed the number. So here, there are no clutter elements. There's 1 here, 3 here, and 6 here. And so in red is the stimulus in isolation. This is 1, 3, and 6. So by the time you add, okay, thank you for coming back. So by the time you have 6 stimuli in the surround, the selectivity of the neuron has been completely washed out. So this neuron does not seem to know the orientation of the central stimulus when you add clutter in the back. So this is basic characterization that told us, okay, this is a worthwhile experiment to do. So then we said, okay, how can we manipulate the saliency of the central target? And we tried different things. So here is one where we're changing the color of the central stimulus. So this is our basic experiment where we have a central stimulus and then we add clutter around it. And here is we painted the central stimulus in a contrasting color, but maintained the same color for the clutter. And the idea being that if it is just based on the summary statistics, then this and this should produce similar results. But if there is something about the saliency of the central stimulus, this should rescue the crowding effect. And so here are the responses of one example neuron. So that's the shape presented in isolation, and these are the 8 different orientations of that shape, and we've just rank ordered it based on strength of response. And this is the tuning curve based on average responses. And these are the responses now for the stimulus by itself in a different color. So this neuron doesn't care about color. The responses are essentially identical, regardless of whether that color is there or not. All it cares about is the luminance contrast between the stimulus and the background. Okay, so it's not a color selective neuron. Then we added the clutter around it, and the responses have completely washed out. So this is, again, a neuron that's really susceptible to clutter around it. And when we present the central stimulus, the target stimulus in a different color surrounded by clutter, you can see that the crowding effect is, the shape selectivity essentially is rescued. So this is the shape selectivity for this particular condition, and then in this pale blue is the effect in this condition. Okay, so having the stimulus in a contrasting color really rescues the shape selectivity in this case. And we've seen this also when we've done these experiments with other kinds of manipulations. So here is when your surrounding clutter are all cluttered stimuli are all circles, either of the same size as the central target or of a smaller size. And when you do that experiment as well, so here are the responses to the stimulus in isolation. These are the responses to the small circle and larger circle. And here are the corresponding tuning functions. So in red is the isolated stimulus, and pale gray is the small circles. This darker gray is the larger circle, and the random shapes is here. So again, by manipulating the structure of the surround, you can now you can actually rescue the shape selectivity. Okay, and this is to we also looked at whether we can decode the identity of the stimulus from the population responses. And so this is the decoding performance for the color experiment. So in red is the decoding performance just based on the shape in isolation. So obviously you can decode which orientation it is quite well. And you can do that for the blue stimulus as well. And in black here is when you are surrounded by a clutter. So the performance collapses. This green line shows chance performance. So performance collapses. But when you have the central target, salient by adding color, you see a little bit of an improvement in the decoding performance. Okay, so I won't go through this in the interest of time because I want to get to the next part. And the other question that I set up here is, can you explain the responses in terms of the summary statistics? So we directly tested that question. So basically, we compared the responses to the metamer of this cluttered display. So we basically created another stimulus that had the exact same first and second order statistics as this cluttered display. And we asked, is the response of the neuron to this stimulus, this cluttered display, is it better explained by a metamer which has the matched statistic, or does it look more like the stimulus in isolation? Okay, so we're trying to understand whether is there some other model that can explain these results, or is the metamer model best? Okay, And we did that also for the cluttered display when the clutter is all a bunch of circles. And if you look at the responses here, so the x-axis is the correlation between the target alone condition and this clutter condition. So the responses between the target alone and this condition, and the y-axis is a correlation between these two conditions. So the metamer versus the cluttered display. And there is a slight shift towards, so the dots are shifted towards the x-axis, suggesting that there's a bit more of correlation with target alone than with this metamer. So the metamer doesn't seem to be a good model for these responses. And when you do that with the circle distractor condition, they're shifted even further. So we think that this metamer model is this summary statistics model is not a good model for how V4 neurons are encoding multiple stimuli. Okay, so to summarize this, so what do we think is going on? What we think is going on is that there is some beyond just so let's say these are the visual inputs, and this is the first layer of a convolutional network model, AlexNet, that's a stand-in for the first level processing. So V1, let's say. The output of this, of a V1 computation, then we believe that there is a saliency detection computation beyond that. So basically, you have some sort of a center surround computation that compares the central stimulus with what's around it. So basically, any stimulus with what's around it, anything that stands out from its surround is something that the visual system is interested in. So we believe that somewhere in mid-level processing, there is some mechanism to differentiate between a stimulus and its surround. If it stands out from its surround, then the visual system says, okay, this is something that I need to think about, I need to encode specifically. The rest can be encoded perhaps with a pooled summary statistic model, but things that stand out from the surround are worth paying attention to. And so that's a framework of a model that we have, but that's something, a more concrete model is something that we need to work on. Okay, so to summarize this part of the talk, pooling of image statistics is not a good model for encoding V4. We think that maybe a tune normalization model that enhances salient contours might be more appropriate for our data. We think V4 responses preferentially encode objects and contours that are salient in terms of color and texture, and motion is something that I didn't show today, but we have some data on that as well. And we think, sort of trying to detect salient contours and encoding them may be important for segmenting objects from the background. And so, and building object-based representations, which is something that we think that may be important. Okay, so that's the first part of my talk. I'm going to pause for a second to see if there are any questions, and I will.
So you have a contrast between the description of these neurons in terms of being sensitive to salient objects, and then you said that they also characterize their texture sensitivity. So these seem like, and then you said, so it's not pooling of image statistics, but it is texture. But texture is image statistics. So I'm confused.
Yeah, very good, very good. Good question. So we do think there are neurons. So when you take a population of V4 neurons, there are neurons that are sensitive just purely to shape. They don't care about texture at all. There are neurons that don't care about texture at all, shape at all, care about texture. And then there are neurons in between that care about both. Texture is important both to segment the object. So there is the localized texture of the object itself, and that's important for segmentation. And then there is the homogeneous texture of the background. So it's a tricky difference to make, but texture is important for segmentation and also for characterizing your background. And so I think the visual system probably uses texture in multiple ways, encoding of texture in multiple ways. So for example, to give you a more concrete example, a neuron that's sensitive to shape and to texture wouldn't respond to a homogeneous texture patch. But if I painted texture on a shape, and if I painted different textures on that same shape, it would differentiate between those. And so that would be a neuron that cares about texture, but only in the context of shape, on the surface characteristic of a shape. Does that make sense?
Yeah.
I'm interested, up to now, you're collecting all this data by measuring signals that come from single neurons at a time, right?
Yeah.
What if, when a brain looks at a complex image like this, many different neurons are doing the recognizing in parallel and talking to each other? And what's happening is that some other place in the brain is integrating all of these informations from distinct neurons.
Yeah.
I mean, could that be happening as well?
Yeah, absolutely. So I do think, so in the next part of the talk, I'll tell you a little bit about recording from lots of neurons simultaneously, and also recording from multiple areas simultaneously to try to understand how these neurons talk. But yeah, this is an effort to simplify the problem by studying one neuron at a time, but that's by no means the full answer, and possibly also the wrong answer, because you've simplified the problem so much that it becomes meaningless at that point. Okay, great. So that provides perfect segue to the next part. So one of the things we're also thinking about is how these neurons then contribute to our ability to recognize objects. And we are thinking about this problem in the context of occlusions. So occlusions are everywhere, all around us. And this makes recognition of objects difficult, and yet our visual system is remarkable at recognizing partially occluded objects. And lots of work suggests that recognizing occluded objects is based on communication between brain areas, specifically communication between frontal cortex and visual cortex. And in our hands, we're looking at the communication between prefrontal cortex and area V4, because area V4 is all we think about all the time. So prefrontal cortex is a good choice for this question, because it's thought to be important for executive control of behavior. It's thought to be important for maintenance of working memory. And other studies have shown that PFC is involved in object recognition and categorization tasks. So to address this question, we trained monkeys on a shape discrimination task. So this slide shows a trial sequence. So the fixation spot comes on and a reference stimulus comes on. After a short delay, a second stimulus comes on. And the task of the animal is to report whether these two stimuli are the same or different with a saccade. So if it's the same, the monkey has to say, make a rightward saccade. If they're different, he has to make a leftward saccade. And the second stimulus is partially occluded. And so we're adding occluders on the surface of the stimulus. We randomly position these occluders, and we titrate the difficulty by changing the diameter of the dots. And so, if you're looking, this may seem like, oh, it's not that difficult, but if you're staring at this dot and you have to discriminate something over here, this is quite hard discrimination. And so we characterize the level of difficulty by measuring the fraction of area that's unoccluded. So as this number decreases, the task difficulty increases. And in our first series of experiments, we studied neurons in V4 first and then in prefrontal cortex. And now we're doing simultaneous recordings in both areas. But when we first recorded in V4, we found that many neurons showed a decline in responses. So this black line here is the response to the unoccluded stimulus. The different colors here show increasing levels of occlusion. And if you see, this black line is the response of the unoccluded stimulus. And then as you go from black to blue to down to red, the responses decline gradually. And this is something others have shown, even in IT cortex. But when we recorded from prefrontal cortex, we saw the opposite. Neurons didn't care much at all when the stimulus was unoccluded. But when the stimulus was occluded, the responses shot up. And they were also selective, so responding stronger to one shape, but weaker to another. Okay. We also looked at, and so then when we saw this, we thought, we wonder if this could provide feedback to visual cortex. But we had done these results, these experiments sequentially. So we just went back to our data and looked for dynamics in the responses. And we found that there were many neurons in V4, maybe roughly 30 to 40%, that showed two dynamic bumps in their responses. So two transient peaks. So the first peak, the responses declined with increasing levels of occlusion, but the second peak showed stronger responses to intermediate levels of occlusion. And so we characterized the first and the second peak of this neuron, and we looked at the average activity during the first peak and the second peak. During the first peak, the responses declined gradually with increasing levels of occlusion, but the second peak showed this inverted U. And we saw that effect in many neurons. So we saw an inverted U in many neurons in V4. So, and then we also looked at the timing of the peaks in V4 and PFC, and we found that PFC neurons peaked, the median duration for the peak was around 150 milliseconds. In V4, the first peak was earlier than that, and the second peak was later. And so this provided evidence that perhaps there could be a feedback from prefrontal cortex to V4. So, so we wanted to test this directly. We wanted to go in, record simultaneously from prefrontal cortex and V4, and we wanted to perturb the activity of prefrontal cortex neurons to suppress the activity of prefrontal neurons and ask what happens to the second bump, the second transient peak in V4. So to do this experiment, we created a cortical cooling probe. So this is a little metal disc that can sit directly on top of the surface of cortex. It's got two tubes through which we pass chilled ethanol. There are channels cut through this cooling probe through which neuropixel probes can pass through. So these are fine probes with hundreds of contacts. Several of you are using this here. And so you can use this to study responses of hundreds of neurons simultaneously. We're also measuring the temperature of the probe, and also we can put in a thermocouple and measure the temperature of cortex. Okay, and I'll show you some data where we're perturbing the activity of prefrontal cortex, and we're studying neuronal activity both in prefrontal cortex and in V4, and we can correlate that and ask how prefrontal cortex affects V4. Okay, so this here shows our probe temperature. So the probe temperature goes from 30 down all the way down to 10 degrees, and the brain temperature modulates only over about 10 degrees or so. And that modulation is sufficient to actually modulate the activity of neurons. So here is just a representation of the neuropixel probe, and here are some neurons that we're recording from. The blue zones are when the cortex is cooled. The white zones are when the cortex is at normal temperature. And you can see that the activity of many of these neurons are oscillating. Several of them are positively correlated with temperature, so getting suppressed during the cooling phase and excited during the warm phase. But there are other neurons that are negatively correlated, so stronger activity when the cortex is cooled and weaker activity when the cortex is warm. So these blue dots represent neurons that are negatively correlated with temperature, and red dots are positively correlated with temperature. We've done many sessions, so these are all different recording sessions with positive and negative correlated units. And if you look across the prefrontal cortex population, you can see a substantial fraction are positively correlated. So the majority of neurons are actually suppressed when you bring down the temperature of cortex. And right now, this is all ongoing work. We're trying to relate the shape of the waveform, trying to extract shape of waveform from the neuropixel data, and trying to relate that to correlations, trying to see if can we see any relationship between a putative inhibitory neuron and whether it's positively or negatively correlated, et cetera. So next, I'll show you what happens in V4 when we're cooling prefrontal cortex. Here too, we see some examples of modulation of activity. So we're cooling prefrontal cortex, and we're seeing some modulation in V4. It's not as pronounced as in prefrontal cortex, but we see some examples. And in this case, we actually see more negatively correlated neurons. So what we see is neurons in V4, the activity of neurons in V4 actually goes up when prefrontal cortex is suppressed, okay? This is the opposite of what we had made up in our brains. We thought, oh, it's a direct excitatory connection, but in fact, we see that the activity actually goes up in V4. We're still working on this. This is, ongoing work, but we can look at the dynamics of the population. So now we're not looking at single neurons, but we're looking at a population of V4 neurons and a population of prefrontal cortex neurons. And we're saying, okay, can we look at what happens to the shape selectivity? So we're showing two shapes in each session. What happens to the shape selectivity during when you cool prefrontal cortex versus when you're not cooling prefrontal cortex. So this is going to be a busy figure, so I'll walk you through it. What I'm showing you up here is the no occlusion case. So the shapes are completely perfectly visible. And remember, I showed you activity where prefrontal cortex doesn't care much about the unoccluded case. So these red and blue lines up here are the shape selectivity measures for V4. And these here are the shape selectivity measures for prefrontal cortex. Blue is cool, red is warm, prefrontal cortex. It doesn't matter, V4 shape selectivity stays strong regardless of what happens in prefrontal cortex. In prefrontal cortex, shape selectivity goes down a little bit when prefrontal cortex is cooled. Okay, that's expected, but nothing much is happening when it's unoccluded. Now, when the shapes are occluded, here is prefrontal cortex. So this red and this cyan is prefrontal cortex. You see that the shape selectivity has gone down quite a bit in prefrontal cortex. In V4 too, shape selectivity has gone down. So the activity magnitude of V4 itself goes up, but the shape selectivity appears to go down. So we're now thinking that maybe we need to think about a new model for how V4 and prefrontal cortex are actually interacting. And one idea we have is based on rodent models of disinhibition control, where, when prefrontal cortex is active, influence on V4 puts the system more in a recurrent frame, whereas when PFC is not active, then V4 gets into more of a feed forward mode. This is an idea we have. We need more evidence for this, but currently now we're thinking about a disinhibition control model for these data. So just to summarize, we've developed a system where we can actually perturb prefrontal cortex activity, and that gives us a way to look at the interactions between two areas. We think that perturbing prefrontal cortex enhances activity in V4 and maybe suppresses selectivity, and we're thinking about a disinhibition control model to describe this. Okay, with that, I'll just put up a slide of acknowledgements, and thank you all for your attention, and I'll take any questions.
I would like to know your thoughts about the definition of visual features that one would like to study as we go higher in the visual hierarchy and as those concepts become more abstract, and where there might be more interferences with like our human way of seeing things. Especially like the contrast with like then looking at single neuron responses of more abstract features like visual texture or crowding versus population.
So I want to make sure I understand your question. So you're asking the definition of what these mean across subjects or across neurons or across between single neurons and the population, or?
Well, it says that when we study the response of neurons in the primary visual cortex, it's easy to say, oh, this response to a specific spatial frequency or shapes.
Yeah.
But once we go higher in the hierarchy, those visual features are less easily definable. Like for instance, for visual texture.
Yeah.
From my understanding, we still don't have a proper parameterization for visual texture.
Yeah, absolutely. That is a major problem. That is a huge problem we have. And I think we have to, it's hard to know what the definition is, right? And I don't think we have an answer yet. I think the best we can do is keep all our options open and be ready to accept that we were wrong when we've sort of pushed a theory forward, and then other experiments show that that's not a good interpretation of the data. So I would say that we probably need to, it's very hard to have a whole bunch of stimuli and test all stimuli in one experiment. And so we end up with subsets of stimuli across different experiments in different labs. And the best way to bring all of that together is probably by testing models. So have some candidate models and bring all of the data to bear on testing these models that define what the feature might be, so that you can test across data sets and validate your models. So yeah, we need to do more of model building and validation as we go forward, I think. Yeah, it's a good question. Yeah.
Could you elaborate a little on the temperature experiment and what was the expectation, I suppose? If I understand, by dropping the temperature, you expected the population of neurons to get suppressed, but then you got excited.
So we had a very simplistic expectation, right? So we said, okay, you have two peaks in V4. You have one peak at an intervening type point in PFC. So we thought, well, maybe that peak was influencing the second peak. So if I bring this down, maybe the second peak will go away, right? And that's not what we're seeing, right? We're seeing that, in fact, the activity is going up, but the selectivity has gone down. So it seems that the presence of normal activity in PFC actually reduces activity in V4, but increases, somehow contributes to its shape selectivity. How this is happening is something we don't know. We have to do more analyses, and we're in the process of where we have our data from the second animal at this point, and so we're working on analyzing that. But yeah. So we're going to revise our theory based on the data. Yeah.
And maybe further excited by the drop of temperature? Is that something that happens in general, or?
So we were surprised by it, but it now seems that Tatiana Pasternak has done a similar experiment. She did it only in one animal in MT, and so suppressing prefrontal cortex and looking at activity in MT, and she found the same thing, enhanced activity in MT. So it seems like there is something there, but I don't know of anybody else who's done this experiment. So yeah.
Any other questions? If not, let's thank Anitha.
Thank you.